welcome to the next edition of the Science Education Series. This time I am with Dr. Laura Brutscher, who is a research scientist here at BioCat Microbials. Thanks for being with us today, Laura.
Thank you for having me.
Absolutely. So I'll just jump right in. Can you give us a rundown of your background?
Sure. So I have been a research scientist at BioCat Microbials for roughly 2 years. I have a PhD in microbiology, and in my training I've utilized microbiology tools such as RNA-seq, PCR, metagenomics, and whole genome sequencing. And I've used these tools to answer various biological questions in organisms from bacteria, viruses, to even insects such as honeybees. At BioCat Microbials, I've brought my love of next-generation sequencing to really assess our bacillus strains for both genes associated with probiotic traits, as well as genes that may be associated with deleterious traits. And that's a very important aspect because we want to ensure that our strains are going to be safe for use as a dietary supplement or as a direct-fed microbial for use in animal feed.
Last year, your team published safety data for BioCat Microbials' newest bacillus strain. Could you elaborate on that, and especially the importance of ensuring the safety of strains before they're put onto market?
Sure. So early last year, we did publish our work examining the in silico safety as well as the in vitro safety of OPTIBIOME BS50, which is a Bacillus subtilis strain. It's important to assess the genome of a probiotic to ensure that it doesn't encode for any toxins or antimicrobial resistance genes. So for example, some species of bacillus, such as Bacillus cereus, encode toxins that can cause severe gastrointestinal disease. Generally, we are not concerned about Bacillus subtilis in terms of encoding any bacillus toxins, but it's important that we do our due diligence. And so we were able to show that BS50, which is a Bacillus subtilis strain, does not encode for any of these toxins that are known to be found in Bacillus cereus. We're also concerned about the presence of antimicrobial resistance genes, as well as their ability to be transferred to other bacteria in the gut, including pathogenic species. Because in that circumstance, we would not want any pathogenic species to obtain antimicrobial resistance and be hard to treat with antibiotics later down the road. So antimicrobial resistance genes can be transferred if they're present on a plasmid or if they're surrounded by genetic mobile elements. And so when we were looking at the BS50 genome, we were not only looking for the presence of antimicrobial resistance genes, but whether they had these traits that could enable their transfer to other bacteria.
And can you tell us more about that strain, OPTIBIOME BS50, and more specifically, how does it benefit gut health?
Yes, so we were able to establish the clinical efficacy and the safety of OPTIBIOME BS50 through a randomized, double-blind, placebo-controlled trial. And this trial included 76 healthy adults. So participants in the trial were supplemented for 6 weeks on a daily basis. Those receiving probiotic received 2 billion CFU per day. Participants were asked to fill out a daily questionnaire on their gastrointestinal symptoms before and during supplementation. And what we found is that after 6 weeks, twice as many of the participants that consumed probiotic had improvements in combined scores for burping, bloating, and flatulence. And so while we're still exploring the potential modes of action of BS50, there are many ways that Bacillus probiotics in general are thought to benefit gut health. So one of them is regulation of the immune system. Another aspect is the secretion of antimicrobial compounds, and that could benefit the gut by reducing the presence of pathogenic organisms. Another major aspect of the potential benefit of a Bacillus probiotic is the secretion of extracellular enzymes. And so that includes lipases, enzymes involved in breaking down fats, amylases, which are involved in carbohydrate breakdown, as well as proteases, which are involved in protein breakdown. And so the culmination of all these extracellular enzymes could aid digestion in the human gut. Another aspect is modulation of the microbiome. And we actually have some really interesting in vitro data to support that BS50 could be doing this in the human gut. What we found is that cell-free supernatants collected from a BS50 culture are able to enhance the growth of Bifidobacterium adolescentis in vitro. Bifidobacterium adolescentis is one of the most common Bifidobacterium species in the adult human microbiome and is typically more prevalent in healthy individuals than it is in diseased individuals. There are a lot of interesting avenues in which we can further explore the mode of action for BS50.